Hello. I would like to thank the speakers for the opportunity to discuss my research. My name is Donahue, and I am a third-year graduate student at the University of Notre Dame, working in Dr. Catherine White's lab. My thesis project focuses on the development of optogenetic tools to spatiotemporally manipulate intercellular pH within single cells. This is because we're interested in studying how intercellular pH affects and drives cell behaviors. In normal cells, transient changes in intercellular pH, which I'll refer to as pHI from now on, regulate cell adhesion, migration, cytoskeleton remodeling, proliferation, and apoptosis. However, in disease states such as cancer, this pHI can become dysregulated, where it's constitutively increased. This increased pHI is linked to increased invasion, metastasis, proliferation, and the evasion of apoptosis. However, all of these links have been shown at the population level. It is currently unknown if pHI is a sufficient or necessary driver of cell behaviors at the single cell level. This is because we currently don't have the tools available to answer these questions. At present, all the tools available to us to manipulate intercellular pH lack spatiotemporal control, are damaging to the cell, and are irreversible. This limits our ability to investigate how pHI drives critical cell behaviors at the single cell level. In this work, we present Archaerhodopsin, which I'll refer to as ArchT, as an optogenetic tool to manipulate intercellular pH. As you can see on the right here, it's an outward proton pump that is activated by 561 nanometer light. We will pair Archae with the mCherry-fluorine Biosensor. mCherry is pH insensitive, while fluorine is pH sensitive. This allows us to calculate and monitor intercellular pH changes at the single cell level. So, how this would work is we would take a snapshot of a field of view of interest, look at the ratio of fluorine to mCherry. We would then standardize with nigericin, a protanophore to two known pHs, 6.5 and 7.6, for example. Once we have that, again, we take a capture of the field of view, look at the ratio of fluorine to mCherry. Once we have that, we can create a calibration curve and back calculate the intercellular pH for individual single cells. So that's how we're going to manipulate and monitor intercellular pH changes in our experiments. And now I want to walk through how we're actually going to set up these experiments. So first, we identify a cell of interest. We draw this stimulation region of interest, shown as a red box here. We then activate Archae only within this region of interest using 561 nanometer light. We can then monitor the change in fluorescence intensity of fluorine. As the intercellular pH of the cell increases, fluorine intensity should also increase. So here's what it looks like in actual cells. So these are retinal pigment epithelial cells, or RPE cells. These are expressing both Archae and mCherry-fluorine. And you're looking at this in inverse mono display. So here, the darker the pixel, the higher the fluorine intensity. And as I play this video, I want you to notice that we are activating Archae only within this red region of interest. And you'll see here that the fluorine intensity is getting darker over the course of the 2.5 minute period. This is indicative of the pHI of the cell increasing, which is what we would expect if Archae is successfully being used to increase the intercellular pH of the cell. So, we then used Archae in more cells and quantified it, and that's what I'm showing you here. So you're looking at delta fluorine over time, and what you'll see here is Archae stimulated cells is shown in red, and our control cells are shown in the other colors. In this case, we have cells expressing only the mCherry-fluorine biosensor, shown in orange and light gray, and cells that express ArchT but are not stimulated in this darker gray. And what you'll notice here is that all of these controls don't really have an increase in fluorine intensity over time. However, our ArchT stimulated cells do have an increase in fluorine intensity over time, which is consistent with an increase in intercellular pH. So this is just another way of looking at this quantification. This is quantified at 30 seconds. However, this trend does hold over the course of the 2.5-minute experiment. And again, what you'll notice is that only our ArchT stimulated cells have this increase in intercellular pH. We quantified this, and at 30 seconds, they are experiencing an increase in intercellular pH of around 0.14 pH units. And at the end of the experiment, at 2.5 minutes, they're experiencing an increase of pHi around 0.28 pH units. This is consistent with pHi increases seen at the population level during normal cell migration assays. This is the first spatiotemporal tool that allows us to manipulate and investigate how pHi is driving single-cell behaviors. So now that we've shown that we can use ArchT to increase the intercellular pH, we want to investigate critical cell biology questions. In this case, we chose to look at cytoskeleton remodeling and membrane ruffling. So I want to go through how we're setting up these experiments. So once again, we identify a single cell of interest. We draw our stimulation region of interest, shown here as a red box. We stimulate ArchT only within the stimulation region of interest with 561 nanometer light. And then previous literature has suggested two hypotheses. The first being that upon undergoing increased pHi, the cell will undergo global membrane protrusions and cytoskeleton remodeling, shown on the top here. The second is that upon undergoing increased intercellular pH, the cell is going to have a localized response. So in that case, we would only see localized membrane protrusions and cytoskeleton remodeling. We can apply this novel tool to study single-cell behaviors such as membrane ruffling and cytoskeleton remodeling. As I play this video, you'll notice that, first off, this is a 3T3 cell expressing ArchT and mCherry-fluorine. You'll notice that upon stimulation only in the red stimulation region, there's an initial retraction of the membrane, but at about 60 seconds, we have induced membrane protrusions and cytoskeleton remodeling. You'll also notice that these induced protrusions are not seen on the other sides of the cell, and this induction of membrane protrusions in or near the stimulation region continues for the course of the 2.5-minute experiment. So we did these membrane protrusion experiments in some additional cells, which I'm showing here. In this case, the brighter the intensity, the more alkaline the pHi. Up top, I am showing control cells. These are cells expressing only the mCherry-fluorine biosensor. When I stimulate these cells, you'll notice that there are no inductions of membrane protrusions over the course of the experiment. However, in ArchT-expressing cells, when I stimulate and activate ArchT, there are these induced protrusions caused by increased intercellular pH over the course of the 2.5-minute experiment. We quantified these membrane protrusions by looking at a change in area over the course of the stimulation experiment. Shown in red here is the ArchT-stimulated cell, and you'll notice that there is an increase in total area of the cell, while this control cell does not experience an increase. We quantified this over a number of cells, and on average, the ArchT cells have an increase of around 36 microns squared, while the control-stimulated cells have a decrease in area of around -10 microns squared. These data suggest that the localized membrane protrusions are induced by the increased pHi that's caused by ArchT activation. With this tool, we have shown that we can use ArchT to increase pHi in a spatiotemporally defined manner. We can use it to investigate pH-dependent biological questions such as cytoskeleton remodeling and membrane ruffling. This has a wide variety of implications in cancer cell behaviors, including migration and metastasis. However, we are continuing to investigate other cell biology questions and optimize ArchT to look at longer timescale biological processes. In this case, we want to investigate cell polarization. This occurs over the timescale of around a half hour to an hour. And in this experimental setup, we would identify a cell of interest, stimulate ArchT, wait, stimulate ArchT, wait, stimulate ArchT, and so forth over the course of an hour, half hour to an hour. And what we see in this data is that the ArchT, when we stimulate ArchT, it gives a consistent, reproducible, robust response over the course of a half hour. This shows promise in us investigating the role of increased PHI in driving single-cell cell polarization. With that, I would like to thank you for your attention, the members of my thesis committee, my lab, specifically Michael Soroki, who is a fantastic undergrad who worked with me on this project. He helped with both data acquisition and analysis. I'd also like to thank the lab's funding, specifically the AD&T organization, who funded this project through a summer graduate fellowship. Thank you very much.